Welcome, ladies and gentlemen, to the latest webinar in the series of Pharm-Olam webinars on key clinical research topics. The subject matter of today's webinar is antibiotic drug development. In the last decade or so, the number of new antibiotic approvals has dropped successively each year. The FDA has changed rules of approval process, but even so, a number of the large pharma have moved from infectious disease research into oncology. Nowadays, MRSA has become a household name, and the demand for new antibiotics cannot be understated. My name is Yamin Khan, also known as Mo. I'm the Executive Vice President for Clinical Development at Pharm-Olam, and I'm based in Ascot in our European headquarters in the UK. Today, I'm joined by our subject expert, Dr. Slobodan Ilic. Dr. Ilic is based in Serbia. He's MD of School of Medicine from University of Belgrade and has been working with Pharm-Olam for the last 12 years. He currently leads the Pharm-Olam Anti-Infectives Business Unit and has extensive experience in conducting antibiotic clinical research, including ICU and pediatric trials, over the last 8 years. He has overseen phase 3 programs in HAP, VAP, CAP, AB, SSSI, UTI clinical trials. In fact, he has worked on 4 out of the 6 newly approved antibiotics of recent times. Before we get into Pharm-Olam's experience and comments on clinical trials in antibiotic research, I would like to briefly introduce you to Pharm-Olam. Pharm-Olam is a 22-year-old full-service global CRO with 29 offices covering 40 countries across Europe, Asia, Africa, and the Americas. We are a mid-sized CRO, privately owned, that prides itself in offering a fast, flexible, and a high-quality product. Over the last several years, Pharm-Olam has conducted over 50 clinical trials in the anti-infective therapeutic area across 22 indications, including abdominal infection, cellulitis, hepatitis, influenza, HAP, CAP, VAP, RSV, respiratory and urinary tract infections. We have recruited 13,000 patients from 1,400 sites across 46 countries around the globe. Currently, we have around 380 staff with experience of conducting clinical trials in the therapeutic area. During today's presentation, we will provide you with an overview of the current antibiotic pipeline, provide tips on protocol and study design, describe the latest regulatory requirements, and sum up with a few conclusions. This will be followed by a short Q&A session. This leads to our first poll question. You will see the questions and the options on the right-hand side of your screen. So the question is, what are the biggest challenges you face in antibiotic drug development? Is it A, slow patient enrollment into the studies? Is it B, the challenges in the variety of standards of care across different countries and different regions? Is it C, meeting the current regulatory requirements and also the different types of regulatory requirements from the different authorities, such as the EMEA and the FDA? Or D, other? Please go ahead and click your answer. You will have 15 to 20 seconds to select your answer. Okay, we will now end the question for the poll. And Dr. Ilic will now take on and provide some commentary on the answer to your question and with the following presentation. Go ahead, Dr. Ilic.
Thank you, Mo. Thanks for these responses, and I must comment first that what you picked here is actually exactly what we expected. So the question that we raised came directly from our experience, and although we could discuss whether the slow patient recruitment, which is usually the biggest challenge in any clinical study, is really the biggest challenge in antibiotic development. I absolutely agree, and let me reiterate one more time, this is something that we expected, that meeting regulatory requirements is probably nowadays the biggest challenge we are facing in antibiotic development. By saying this, I don't mean meeting all the agency's requirements, but also keeping up with local requirements, with the requirements of different countries and their both regulatory and scientific environment. I will expand on these topics during this short webinar, and as beginning of the webinar, I chose to speak about first a little bit about public awareness related to antibiotic development. And then I will say something in addition to study design, to some important issues and obstacles and hurdles, pitfalls during antibiotic development. And finally, I will stress on some tips that are coming directly from our experience of doing several clinical studies in the last couple of years in different indications in antibiotic development. So I would like the host to enable me to change the slides because I cannot do it at this moment. Okay, so I'm going to start with public awareness about regarding problems we are facing in antibiotics development. Not so long ago, we thought that the battle against bacteria and against pathogens is won when the pioneers in antibiotic development discovered substances that can kill bacteria. Several decades later, we were proved not wrong, but probably terribly wrong, because as it happens, bacteria develop some defense mechanisms that we nowadays call resistance. So, to raise public awareness about what's going on in the field of infectious disease, especially in the field of bacterial resistance, is very important. So on this slide, you can see some scary or some even terrifying headlines from the news saying that the battle against bacteria is not really won, saying that there are bugs around us resistant to currently available treatments. And finally, raising public awareness about this and, of course, the need for funding and for investing money in antibiotic research. Why is it so difficult? Well, it is common knowledge that big pharma actually need around 800 million annually to recoup for their research and development costs. It's about 100 million for small biotech companies. So this is a huge amount of money. And the inevitable question for big pharma companies was why we should invest in such an expensive development when we can get greater profit on drugs that can be used regularly without losing effectiveness, unlike the antibiotics. So some of the major players in this area left the playground and it's been going on in the last 2 or 3 decades. So you shouldn't be surprised when I tell you that the last newly discovered class of antibiotics happened actually in 1987. And this was the case of oxazolidinone, first in class of oxazolidinone antibiotics, linezolid, developed by Pfizer. So, if I may move to the next slide, please, because I cannot control this presentation, obviously. So, with the funding of antibiotics research, there are some interesting incentives and supports that have been going on for the last 10 years or so. I would like to point out, for instance, the initiative from Infectious Disease Society of America, which is called 10 by 20. What it means actually is that it is absolutely necessary and expected and set up as a goal to develop 10 new antibiotics by the year of 2020. So, in addition to that, a lot of interesting things happened, starting from the GAIN Act, which is the FDA Safety and Innovation Act, set up actually to encourage research into new antibacterials. So this started almost a couple of years ago, and what the FDA is assigning nowadays is qualified infectious disease product or QIDP framework, which is based on this GAIN Act. Apart from that, you can also see a lot of incentives and a lot of support from BARDA, from Innovative Medicines Initiative, and probably the most interesting one would be, and very important to mention, the initiative and program called New Drugs for Bed Bugs. So it is, of course, evident that we are surrounded with more and more resistant bacteria, and we need some serious programs and some serious fundings to fight this. So under this New Drugs for Bed Bugs program, you can see different aspects and different approaches. Approach related to drug discovery, approach related to drug development, to economics and stewardship, and basically, let me give you an example. Translocation program is actually investigating how to transport antibiotics into bacteria. There's another program called COMBACT, which focuses on the design, on the implementation of more efficient clinical trials. Finally, there is the NABLE program, which is all about creating a drug discovery platform to fast-track this development of promising molecules. And this is all about the discovery and development of new drugs combating gram-negative infections. So let me switch to antibiotics pipeline, and let me try to explain what is going on in terms of clinical trials, in terms of recent development, and of course, in terms of promising molecules that we are actually witnessing at this very moment. So looking at the global data, you can see different parts of this cake explaining what are different phases of the research in antibiotics. So you can see that the most of the research is 41% in phase 2, 25% in phase 3. On the other hand, in the total numbers, when you look at the clinicaltrials.gov, and these are data pulled out on September 27th this year, you can see that in terms of anti-infective development, you have around 2,400 So is this really correct? Is this really true? Well, let's go to the next slide and see. Well, when you see that there are 2,400 clinical trials in antibiotics area, you would expect a lot of antibiotics approved for clinical use. However, since year 2014, only 6 antibiotics, 6 new molecules or 6 new combinations or whatever are actually approved as a novel medications. I have listed them here, and it's only 6. I had the pleasure to work on 4 out of these 6, and what is interesting about all these drugs is that we're not looking here into new classes of antibiotics. This is something some people would call new patches for old problems. So the holy grail of antibiotic development, which would be a new class of antibiotic, is still not even close to the market. As you can see, the first 3 drugs are products developed and labeled for skin infections. So ABSSSI stands for acute bacterial skin and skin structure infections. Both dalbavancin and oritavancin are not new molecules, and their pathway was very, very interesting. For instance, dalbavancin was a molecule that was identified in 1987, but failed initial research, and finally through the company called Durata Pharmaceuticals got its label for ABSSSI. And when you see that the antibiotic is developed for ABSSSI, it tells you that the antibiotic is developed for gram-positive pathogens. And ABSSSI are predominantly caused by Staph aureus, very frequently methicillin-resistant Staph aureus or Strep pyogenes. On the other hand, there is ceftobiprole, which is a cephalosporin antibiotic, and initially developed as the first cephalosporin, actually the first generation 3 cephalosporin active against MRSA, and it was actually developed at the same time as ceftaroline. However, at one point of the development, ceftobiprole, which was actually developed by J&J, failed to get the approval from FDA due to some severe errors in a drug development. So the last 2 products, ceftazidime-avibactam and ceftolozane tazobactam, are also cephalosporins, but linked to beta-lactamase inhibitors, and currently labeled for abdominal and urinary tract infections, and at this very moment are being in the research for nosocomial pneumonia. Next one, please. So I'm showing you here the table with antibiotics in the clinical development, and we have 3 slides showing this. There are 27 molecules listed here, but again, what I'm trying to emphasize, looking at the table of 27 molecules, you will see that there are no new classes in the development. Also, what is important is when you look at the tables and you basically look how these molecules are classified. So basically, if you can see very few molecules which are expected to be active against gram-negative so-called escape pathogens. Escape is an acronym for Enterococcus faecium, Staph aureus, Klebsiella pneumonia, Acinetobacter, Pseudomonas, and Enterobacter. So nothing new. It's just old classes and new molecules, and some of them active against gram-negative escape pathogens. If we had this webinar several years ago, we would probably be talking about MRSA and emerging threats from community-acquired MRSA, but this actually changed, and in the last several years, what we are facing as a primary problem is gram-negative bugs. Next one, please. So, what are the current trends and what we have to be aware of when we at least start thinking about developing antibacterial drugs? So, you have to be aware of one historical fact, and that is a low rate of success in ID drugs. Only 1 out of 5 comes to the market. And as I showed you in this previous table, it has recently become quite surprisingly competitive environment. It wasn't the case 15 years ago. And what we have to be aware of also is the situation of a dog chasing its tail. So in other words, once you start the antibiotic development, it takes time to get it to the market. And don't be surprised that by the time you get the drug on the market, the pathogen that you were developing the drug against is no longer a burning threat. So it's the case for MRSA. In 2011, we had the emerging threat with community-acquired MRSA. By the time Tadizolid came to the market, which was June 2014, we can easily say MRSA was no longer the primary problem. Primary problem, obviously, is gram-negative resistance. Now, what you have to focus on is, of course, bacterial resistance. So there is a statement from WHO that antimicrobial resistance is an increasingly serious threat to global public health and requires action across all government sectors and societies. You have to be aware what are emerging threats. Nowadays, we are trying to struggle against particularly nasty pathogens called carbapenem-resistant gram-negative pathogens, predominantly Enterobacteriaceae, Pseudomonas, and Acinetobacter. At the end of the day, you also have to be aware what are the novel approaches in antibacterials. So it's not necessary that in future bacterial infections are going to be treated by antibiotics. Several companies have started with the development of monoclonal antibodies targeting specific epitopes on certain bacteria. Nowadays, you can see that with MRSA, with Pseudomonas, with C. difficile as well. So, I'm giving you here just the example of antibiotic resistance of Klebsiella pneumonia. Klebsiella pneumonia belongs to the class of Enterobacteriaceae, and what's becoming actually a big problem is the carbapenem resistance of these bugs. These bugs can create something that is called KPC, which is an enzyme that basically deactivates carbapenem antibiotics. So carbapenems for us were like the last resort for the struggle against gram-negatives. So if we lose them, we're going to be in trouble. And interestingly, when you look at this slide, I only showed you here Klebsiella pneumonia and the resistance of Klebsiella. So the more eastern you go, the bigger problem you have with resistance. So you can see India, and you can see carbapenem resistance in India or Romania or Serbia. It's really terrifying. So in the last couple of years, our clinicians are facing big problems, nothing to give to the patients to treat their infections. And basically what they're forced to do is to go back to some old and almost forgotten drugs like colistin, which is in a way good drug for certain pathogens, but again, its toxicity is not really favorable, and we really have a lack of understanding how to dose it and for how long to treat the patients with it. Okay, next one, please. Just a couple of words about monoclonal antibodies and potential advantages behind this completely novel approach. So there are several advantages. The first, of course, would be the safety, as the monoclonal antibodies are considered to be pathogen specific and would not cause any drug interactions with other small molecules. It is a specific treatment targeting specific epitope on bacteria and not causing cross-resistant, and it shouldn't have any effect on the beneficial microbiome, which is happening with treatment with regular antibiotics. Antibodies have long half-life, which gives us the opportunity not to treat patients with them, but also to have some kind of prophylactic treatment for the patient. And of course, antibodies could be added to antibiotic treatment, so there would be no resistance to antibodies. And they are easily complementary with antibiotics. And of course, what we hope to see is the reduction of inflammatory response, like systemic inflammatory response syndrome in septic patients, which is a huge problem for patients with severe infections. Next one, please. Couple of words about the general obstacles in antibiotic research. So, developing antibiotics is a complex business. Of course, having bacteria with such a developed, highly effective defense mechanisms and giving them the opportunity to mutate over time, to be resistant to many of the medicines available to treat them. So it's probably one of the biggest problems that you have to solve with antibiotic research. It's a complex science. It's a complex from both parts, something that I call scientific and, of course, something that I call operational part. Protocols of clinical trials in antibiotic research are pretty complicated, and the processes within clinical trial research in antibiotics are complicated. Again, you have to apply several diagnostic procedures within the antibiotics. If you're developing the drug, you have to think of an X-ray, you have to think about microbiology lab, you have to think about even fast diagnostics, if the speed tests are available. Can you translate results from the clinical trials to a real world? And then it's a challenging commercial environment. Some data do suggest that the time frame for development of the antibiotics from the research lab to the market is around 12 years. That's terrifying. Next one, please. Couple of words about the protocol and about study design in antibiotic research. So several things you have to bear in mind, and of course there are some specific things in antibiotic research, some that are not so specific. Now, when I talk about the antibiotic development, I tend to talk about later stages of development, efficacy trials, and trials that you are actually undergoing through to submit NDA and finally to register the drug. But specifically in certain indications, early phases of clinical trials in antibiotic development cannot be underestimated or forgotten. So I'm giving you here the example of specific trials looking for QTC prolongation, or trials actually giving the answer to the question, can the drug be used for treating pneumonia, which is one of the biggest challenges in antibiotic development. So probably some of you know what happened to daptomycin, which appeared to be inactivated by a lung surfactant based on the results from phase 3 studies. So it could all be avoided just by performing a simple DAL phase 1 study and taking epithelium lining fluid from the patients, from healthy volunteers actually, and looking if the drug penetrates into the lungs. There are two major ways to design the study. You either go for non-inferiority, which is most of the studies I worked on, or superiority trials. If the indication you are investigating actually has some available treatments that can actually cure this specific condition, you would go for non-inferiority. You would set up the margin, which is usually around 10%, and try with your efficacy results to remain within this margin. Usually, in the later stages, we are looking into randomized double-blind trials and with, of course, active comparators. What is very specific for anti-infective antibiotic trials is that those trials are very short in screening, 24 hours for screening, and then you start the treatment. So in other words, everything that needs to be done, eligibility that needs to be confirmed, has to be done within a very, very short time frame. And when you design the study, make sure you identify what are the visits that are of outstanding importance. And usually in the antibiotic trials, you have very specific visits where you assess primary endpoint, primary efficacy endpoint, and you really have to make sure you don't miss this, either in terms of timing or in terms of procedure that is required. You have to be, of course, very careful when you choose your comparator, as well as when you choose your endpoints, which should match both agency requirements, so what regulatory agencies expect from you in terms of data. On the other hand, you have to make sure you follow what are the scientific and clinicians' expectations. Finally, when you develop the antibiotic, you have to make sure that you think about pediatric considerations and about the use of antibiotics, not just in adults, but also in pediatric and other vulnerable populations. Now, this is a typical design in antibiotic studies. Patients starting with finding ICF, and then screening period, which is very, very short, usually only 24 hours, followed by randomization and active treatment. Usually, antibiotic studies have end of treatment visit, test of cure visit, and the late follow-up visit. In the old design and conservative design, test of cure visit was always the visit when the primary endpoint was assessed. And this is basically where investigators are assessing whether the drug works or not. However, in the last couple of years, FDA has changed this approach and moved this primary endpoint towards some more objective assessment. And I will tell you this a little bit later when I come to regulatory requirements. Next one, please. So, make sure that you are aware that your protocol is a living organism. So once you start the study with the initial protocol, it's not unusual to change this protocol. I have provided here several examples from our previous studies where we realized in the middle of the study or after we initiated the recruitment that some of the criteria, some of requirements in terms of inclusion of the patients are simply out of date and unnecessary, like Apache score in ventilator nosocomial pneumonia studies. Sometimes you may even change duration of treatment, or if you get the new data about potential interactions, change the list of prohibited or allowed concomitant medications. And of course, you may also, during the study, change the guidance on how to deal with the patients who have certain pathogens isolated at the baseline. Next one, please. So, when you write the protocol, probably inclusion and exclusion criteria are the most critical part. You go to your KOLs, and they will give you definitely their opinion and whatever they think is necessary to be done. But make sure that KOL written protocols are not always feasible. You have to also match the real life situation. So try to match enrollment criteria to current regulatory and industry guidelines, but don't let those criteria kill your recruitment, because it may happen. Just to basically narrow the population of the patients to the point that you have no one to recruit in the study. Always listen to the CRO with experience in the indication you are developing the drug, because this is where you're going to get some very valuable and meaningful guidelines. And in terms of logistics, your planning is absolutely crucial. You go where the epidemiology takes you. So, for instance, if you are after MRSA, you will target the regions with a known high prevalence of MRSA. You will also look for the sites with a microbiology lab that is available 24/7. How else will you be able to run the study? And some big pharma companies are actually having studies with sites that are being closed one after the other because they didn't think properly about setting up microbiology lab and the processes around microbiology. Use of rapid diagnostics, turnaround of data, and of course, training of the staff and availability of the staff. Let me remind you, 24 hours of screening and several days of very intensive treatment. So this is demanding, and this requires a certain number of people involved at the site level, and of course, being available all the time. Next one. For certain studies, I have to point out that informed consent is a big issue. So you have, of course, 24 hours to obtain consent and confirm that the patients are eligible. If you're conducting ICU study, you would be probably dealing with a high percentage, if not 99%, of unconscious patients. You're doing study in nosocomial pneumonia or in sepsis, where most of these patients, if not all, are intubated. So think about the caregivers, think about the local regulations when it comes to signing consent by a legally authorized representative, and know local laws and requirements, of course. You're not going to go to the country where it is either impossible to sign the consent by someone else, or it takes too long to get it. Next one, please. Finally, I'm going to just give you a brief overview of some interesting regulatory requirements. I'm not going to talk about local country-specific regulatory requirements for antibiotic development. What I'm going to try to do is to give you some examples of how the agencies can dictate protocol development and how careful you have to be when you set up your primary endpoints when you design the study. So it was common knowledge that all antibiotic studies are basically ending with a test of cure visit where investigator provides his opinion and his assessment of clinical outcome and patient's response to the treatment. So in a way, that was a kind of subjective endpoint. So in the search of objectivity, FDA has completely changed this paradigm. So, for instance, in studies where you are enrolling patients who are intubated in ICU studies, primary endpoint for FDA is mortality. So, when you look into microbiology, into radiographic endpoints, into clinical endpoints, so nothing is objective enough or as objective as a mortality. Some clinicians do not like that, and I do understand them because I come from the ICU environment. However, this is how it is. So the example of nosocomial pneumonia study is that you're basically developing the study with two co-primary endpoints. The first is 28 days all-cause mortality for FDA, and the second is therapeutic response at the test of cure visit. So, in other words, in Europe, we're still around test of cure visit and testing clinical success of the treatment at the test of cure visit. The other example, for instance, is the skin infection study. So instead of looking at therapeutic response at the test of cure, FDA now wants to see early response to the treatment, which is 48 to 72 hours after the commencement of the treatment. So therapeutic response at the test of cure still remains a secondary endpoint for FDA, although a primary for EMA. And this is definitely something to think of when you're developing the protocol and when you train your investigators. So there are two important moments in the clinical study that you don't want to be missed. So the tip is definitely review the guidelines and make sure you follow them and you please all the agencies that you need to please and make happy with your data. Now there are other tips which are somehow not specific for antibiotic studies, but again important to mention. For instance, site selection issues with starting from feasibility, from turnovers, from resourcing. This is very important. And I have seen so many sites closed or non-recruiting and being unsuccessful in a clinical trial because of these actually basic requirements. So when you do feasibility, make sure you do a detailed feasibility. Don't just send a synopsis to an investigator because there is a high chance they wouldn't read it at all. They will look for an indication and say, okay, I want to participate or I'm not going to participate. But if they say yes, make sure you are aware of their logistics, of their labs availability, especially about their microbiology labs. I have seen the studies in antibiotic development where microbiology lab was not aware of the study at the site, and that's a road to disaster. Of course, regulatory approvals and overall regulatory issues are common in any clinical study. They may take some time, and you may, of course, face the issues with contracts. You may face the issues with drug supplies as well. So be prepared for that. You may be in a situation that you cannot basically you cannot get enough sufficient amount of comparator to the site, or of course, if you're buying it locally, make sure you do it in a way that the site is always adequately supplied. And always make sure you train the team at the site initiation visit. Try to gather as many people as possible because this is something crucial for them. Patient recruitment, loss of motivation, consenting issues, bad feasibility results. Investigators in any clinical study tend to overpromise. Be careful with that. And finally, try to help them by, for instance, developing training materials for the site. You develop laminated sheets for inclusion-exclusion criteria, things like that. Always welcome at the site level. Make them aware that there is a clinical study going on in the ER if you are recruiting patients from the ER, like in community-acquired pneumonia studies or skin infection studies or UTIs. And finally, as a conclusion, it's obvious, it's definite that we need novel antibiotics urgently. We are currently facing some pan-resistant strains of gram-negative bacteria, and these are terrifying. I have seen several in the clinical studies we are conducting so far. And when you see microbiology report stating that the bug is resistant to everything that is available, it really makes you think. So the funding and research facilitation, starting from public awareness, is definitely something that we need. And I must say I'm deeply interested in this novel approach. I'm deeply interested to see what's going to happen with monoclonal antibodies. It was a huge promise in oncology and rheumatology. When you talk to oncologists, they expected more, and I hope that this kind of targeted treatment in the ID environment will be more successful. And then finally, plan your study carefully. Try, again, I'm repeating this all the time. As much as you need to match regulatory requirements, you also need to take into account what the scientific environment is telling you and what the clinicians are telling you. And finally, I'm going to end with a quote from Voltaire saying, "Don't let the perfect be the enemy of good." And this sentence was actually used by the researchers from Duke University, led by Ralph Corey, who were extremely unhappy by the FDA changed paradigm in skin infection studies, and basically they wrote an interesting article and actually called it "Don't let the perfect be the enemy of good." So I thank you for your attention.
Thank you, Dr. Illich, for a very informative presentation today. We are now ready for questions. I know some of you have already submitted some questions using the WebEx interface. Please go ahead and submit any further questions you may have. While you are doing that, I would like to inform everyone that Dr. Illich will be attending the ID Week conference in New Orleans towards the end of the month. And if anybody would like to have a short meeting with him, please send an email to info@pharm-olam.com. So we will now go through some of the questions raised by various people during the presentation. So the first question is, what are the biggest obstacles in antibiotic clinical studies conducted in the ICU?
Okay, so there are several issues with ICU studies. The first and the most important one is actually the severity of the patient's condition. When you are trying to develop antibiotic in the indication, which is basically under ICU and intensive medicine. So you have to first be aware of certain scientific and certain practical obstacles in the ICU medicine. So, starting with a simple thing, as a consenting procedure, you have to make sure that the study you are working on is actually done in a country where it is easy or allowed to consent the patient who is unconscious, who is being intubated, mechanically ventilated, and sedated. Second thing is ICU is the department in the hospital which is basically 24 hours of intensive care. So in order to avoid some stupid or bizarre events, you have to make people aware that there is a clinical study going on and that the patient is actually participating in a clinical study. Otherwise, it may happen to you, and I'm sharing this with from my personal experience, that the patient in the ICU is actually enrolled in the study by the team who worked in the morning. In the afternoon, the second team came into the hospital and gave additional set of antibiotics, additional doses of antibiotics, having no idea that the patient already received study treatment. So it's just a simple example. Second thing, there are assessments in the ICU that are standard for every single antibiotic study. There are a lot of scores that you're going to perform for those patients. Probably Apache score, which is always huge obstacle for investigators. SOFA score, CTIS score. So so many things to think of and to put the patient through, and all of them requiring some serious training. Because if you want to avoid messing up the data, you have to make sure that the patients are assessed adequately, and they will always raise issues, for instance, on how to calculate Glasgow Coma Scale in a patient who is already sedated, or
okay.
How to deal with certain parameters that are changing over time. So these are some more medical issues and scientific issues. But anyway, I hope I provided some at least superficial guidelines on what you need to think of if you go to the ICU.
We have quite a lot of questions coming now, so we have 3 minutes left. So maybe we could go through this fairly quickly, at least some of them. Are there any major differences between conducting clinical research in antibiotics, antivirals, or antimicrobials?
Well, of course, there are big differences in antibiotics and antivirals. I would say that the research in antibiotics and antimycotics are more or less similar. Antivirals are a little bit different. So depending on what you are doing in the antivirals, are you developing a vaccine or some kind of different treatment? It may happen that you are treating some chronic conditions, and the endpoints are different, and study dynamics, as I like to say, is completely different than in antibiotics. In antibiotics, everything is quick, everything is short term, and everything is tied to a very specific and short time frame. And I would say that's the biggest difference.
Thank you. Why is lung penetration important for early phase?
Lung penetration is important because if you're trying to develop a drug for pneumonia, you have to make sure that the drug gets there. So some drugs like vancomycin poorly penetrate into the lungs. So once you treat the patient with this drug and you try to sort out pneumonia or to treat pneumonia, you may need higher doses of the drug to make sure that bacteria that are causing these infections are adequately treated. Some drugs do not get into the lungs. I tried to give you the example of daptomycin. Daptomycin gets inactivated by a fluid that is present in the lungs that is called a lung surfactant. So in order to make sure that your drug is going to get there and going to be in the adequate concentration in the lungs, you perform a very simple study. You give the drug, and then you perform bronchoalveolar lavage. And then what you do is simply measuring concentration of the drug in the fluid you obtain by this method. And then you know, okay, I have the drug in a sufficient amount in the lungs, and I can expect some effectiveness in treating pneumonia. That's why it is important.
Thank you. I appreciate there are a number of questions still unanswered. As we have run out of time, we will follow up each individual with the answer to the questions. I would just like to thank everyone for participating in this webinar today. Thank you, Dr. Illich, for a very informative presentation. Before I say goodbye, please look out for our next webinar on rescue study tips for success to be held on the 10th of November of this year. All that remains for me to say is thank you again for your time and for your attendance. We look forward to speaking to you again. Thank you very much. Goodbye.